Okay, team, listen up. I want to walk you through this single most important document in this lab. Think of it as our Bible, our playbook. It's what ensures the work we do here actually matters. So, welcome aboard. We're really glad you're here. But let's get one thing straight from the jump. Every single discovery we make, every paper we publish, it all rests on one thing: rigorous, obsessive measurement. You see, bad measurements don't just give you bad data, they give you bad science. And that's not something we do here. So here's our game plan. This is the playbook. We're going to start with the big picture, our philosophy on measurement itself. Then we'll drill down level by level. We'll go from the ultimate evolutionary scorecard, Darwinian fitness, down to its building blocks, the life history traits. From there, we'll look at the physical changes, and finally, right down to the nitty-gritty of metabolic shifts. This structure, this is how we make sure our work holds up. Right, Section 1. Why are we so obsessed with this? Well, to really get why we do things the way we do, you kind of have to understand the history of our field, and maybe more importantly, the mistakes people made along the way. If you look back, before the 80s, a lot of the work in this area was honestly a bit of a mess. The techniques were often inconsistent, and things like replication and controls were all over the place. The whole field had to learn the hard way that without super strict, standardized rules, the results you get are basically meaningless. So the rigor we have today, it was born from those early failures. So let's jump to the top of that pyramid. Darwinian fitness. This is it. This is the ultimate scorecard for evolution. But how we actually measure it all hinges on one tiny, very complicated factor: sex. And this slide really shows you two completely different worlds. On one hand, you have asexual microbes like E. coli. Measuring their fitness is beautiful. It's a straight-up race. You can mark the original ancestor and the new evolved guy, throw them in a flask together, and just see who wins. It's simple, direct, elegant. But as soon as sex enters the picture, everything gets way more complicated. We can't do that simple race anymore. We have to use these indirect methods like counting up all the offspring or trying to piece fitness together from all its different parts. Which brings us right to those parts, the life history characters. We're talking survival, reproduction, mortality rates. If fitness is the final score of the game, these are the individual player stats that add up to that score. Now, when we measure viability, juvenile survival, there are two rules that are written in stone around here. Rule number 1, you have to measure it in the exact same environment where you did the evolution. Rule number 2, standardize the density. I can't stress this enough, it is absolutely non-negotiable. Survival rates can swing wildly depending on how crowded things are. If you don't nail down the density, your data just isn't repeatable. Period. And if you think that's tough, what about measuring adult mortality? The scale of the challenge just explodes. To get solid, age-specific data, you can't just follow a handful of them. No, you need cohorts of 2,000 or more. It's a huge investment of resources, but it's what it takes to do it right. So why all the fuss about matching the environment? It all comes down to this one crucial concept: genotype by environment interaction. It's a fancy way of saying that the winning organism in one place might be a total loser in another. If you test your subject in the wrong setting, you can get a completely misleading picture of what's actually going on. And you might be sitting there thinking, okay, sure, but is this just some academic, theoretical problem? Does it really matter that much? Oh, it's not theoretical at all. It is as real as it gets. Take a look at this quote. This study with fruit flies found that when they simply switched the testing environment to match the actual experimental conditions, they got, and I'm quoting here, radically different results. Getting the environment wrong isn't a tiny mistake. It can completely flip your conclusion on its head. And this same level of control applies to everything, especially reproduction. I mean, testing virgin animals when your whole experiment involves mating? That's just wrong-headed. Density, food, how much competition there is, it all has to be locked down and standardized. These aren't just suggestions, They're the price of admission for getting good data. Okay, so we've gone from a big picture score down to the individual stats. Now let's go a level deeper. Let's look under the hood at the physical machine itself. Physiology, morphology, behavior. Because here's the absolute central challenge for us. You see a change in some trait, and you see fitness go up. So, did that trait cause the fitness increase, or was it just along for the ride? Is it a true adaptation, or just a coincidence? How do you tell the difference? Well, this is where experimental evolution becomes this incredibly powerful tool. We can do what's called a focal selection experiment. Instead of just letting nature do its thing, we become the ones doing the selecting. We can pick one specific trait, like, say, flight endurance, and actively select for it. By doing that, we can untangle all the messy correlations and figure out its true contribution to fitness. It's how we get from maybe to yes, this is why. All right, final stop. We are drilling all the way down to the bedrock, the chemical engine of the cell itself. How does evolution actually rewire an organism's metabolism? This table is from the famous long-term evolution experiment, and it's just a perfect snapshot of adaptation in action. Look at this. After thousands and thousands of generations of E. coli, you can literally see which parts of the cellular factory got turned up, like the main power plant, the citrate cycle, and which parts got turned down or even turned off, like building a flagellum. Evolution is the ultimate efficiency expert. If you don't need it, you ditch it. So, wrapping this all up, what's the one thing I want you to take away? It's this. From the 30,000-foot view of fitness, down through life history, down to function, and all the way to the molecules, this precise hierarchical approach to measurement is everything. It's the foundation that turns a cool observation into a solid, reliable discovery. It's the heart of everything we do. We follow this playbook so religiously for one reason: to generate the best, most reliable data on the planet. These methods, all this rigor, it's all in service of one thing: answering the really cool questions. So, the last question I've got is for you. Now that you know how we get the data, what's the next great question you're going to ask with it?